All right, today we're cracking open a scientific cold case. It's a genuine mystery that stumped the smartest scientists for decades, a molecular whodunit that once it was solved, completely changed how we understand life itself.
So, you see, by the 1920s, scientists knew that our hereditary information, the stuff that makes us who we are, was packed away in chromosomes inside our cells. But here's the problem. They had no idea what that information was actually made of. What was the molecule that held the blueprint for life?
Now, in this investigation, there were two main suspects. In one corner, you had proteins. Proteins do everything in the cell. They're incredibly complex, built from 20 different building blocks, and they run the whole show. To most scientists, protein was the obvious culprit. Of course it was. And in the other corner was DNA. DNA just seemed, well, kind of boring. It's just a long, repetitive chain made of only 4 basic parts. It felt way too simple to hold the code for something as complicated as a human being. The smart money was on protein.
But, every great mystery needs a breakthrough. It needs that one clue that just doesn't quite fit. And that first huge clue dropped in 1928, thanks to a British bacteriologist named Frederick Griffith. And let me tell you, it changed everything.
So Griffith was studying the bug that causes pneumonia, Streptococcus pneumoniae. And he noticed it came in 2, let's call them flavors. One was the S strain for smooth. It had this protective coat that let it hide from the immune system, which is what made it so deadly. The other was the R strain for rough. No coat, so it looked rough, and it was totally harmless.
Okay, so Griffith set up a series of really clever experiments with mice. First up, the baseline test. He injected a mouse with the live, deadly S strain. And, just as you'd expect, the mouse got sick and died. That confirms it. The S strain is the killer. Next, the control. He injected a different mouse with the harmless R strain. And, right on cue, the mouse's immune system handled it. No problem. The mouse was perfectly fine. So far, so good. Nothing surprising yet.
For the third experiment, Griffith took that deadly S strain and cooked it, basically. He killed the bacteria with heat. Then he injected those dead bacteria into another mouse. The result? The mouse lived. This proved the bacteria had to be alive and kicking to cause the disease.
Okay, let's recap real quick. The live R strain is harmless. The dead S strain is harmless. So logically, if you mix 2 harmless things together, the result should be harmless too, right? Well, this is where the investigation took an absolutely shocking turn. When Griffith injected a mix of the harmless live R strain and the harmless dead S strain into a mouse, the mouse died.
This result was mind-blowing. When Griffith looked at a blood sample from the dead mouse, he found living, deadly S strain bacteria. What? Somehow, something from the dead S strain cells had been absorbed by the living R strain bacteria, and it gave them the instructions to build that deadly coat. The harmless bacteria had literally been transformed into killers.
Griffith's experiment was a total game changer. It proved that some physical substance could carry genetic information from one organism to another. But it left this giant question mark hanging over the entire scientific community. What on earth was this mysterious transforming principle made of? The case was far from closed.
So, who's going to solve this mystery? Well, the task fell to a new team of scientific detectives. For over a decade, from 1933 all the way to 1944, these 3 guys, Oswald Avery, Colin MacLeod, and Macklin McCarty, worked tirelessly to figure out exactly what Griffith had discovered.
And their approach was just brilliant. It was like a forensic process of elimination. First, they figured out how to isolate all the major molecules, the proteins, the RNA, and the DNA, from those heat-killed S strain bacteria. Then, they used these specific enzymes. Think of them like tiny molecular scissors, to destroy just one of those molecules at a time. The final step was to see if the leftover mixture could still do the magic trick and transform the harmless R strain.
And this table right here lays out their incredible results. So in the first test, they added enzymes that chew up proteins. They mixed that with the R strain and boom, transformation still happened. Okay, so it's not protein. Next, they used enzymes to destroy the RNA. Again, transformation happened. It wasn't RNA either. But then came the moment of truth. They added enzymes that specifically destroyed DNA. They added that to the R strain, and for the first time, nothing. The transformation was completely blocked.
You can't get much clearer than that. This was the smoking gun. The only time the transformation failed was when the DNA was gone. This was the first direct, hard proof that DNA was the molecule carrying the genetic instructions. The transforming principle had a name.
So after more than a decade of incredibly careful work, the verdict was finally in. Even though it seemed way too simple for the job, the evidence was undeniable. DNA, not protein, is the genetic material.
So let's just run through the case file one more time. First, Griffith's incredible experiment showed that some physical stuff, this transforming principle, could pass genetic traits around. Then, Avery, McLeod, and McCarty did the hardcore forensic work. They systematically eliminated the suspects one by one until only a single culprit remained. By showing that the magic only stopped when DNA was destroyed, they proved it. DNA is the molecule of heredity.
Identifying DNA as the genetic material was a huge achievement. It solved one of biology's greatest mysteries. But like all great discoveries, it immediately opened up an even bigger, more profound question. They had figured out the what, but the how was a complete puzzle. How in the world could this one seemingly simple molecule store and transmit the vast, complex instructions for building an entire living thing? Well, that discovery, that's a story for another time.